An odor or odour or fragrance is caused by one or more volatilized chemical compounds, generally at a very low concentration, that humans or other animals perceive by the sense of olfaction. Odors are also commonly called scents, which can refer to both pleasant and unpleasant odors. The terms fragrance and aroma are used primarily by the food and cosmetic industry to describe a pleasant odor, and are sometimes used to refer to perfumes. In contrast, malodor, stench, reek, and stink are used specifically to describe unpleasant odor. The term smell is used for both pleasant and unpleasant odors. In the United Kingdom, odor refers to scents in general. In the United States and for many non-native English speakers around the world, odor generally has a negative connotation, as a synonym for stink. On the other hand, scent or aroma are used by those people to indicate pleasant smells. Basics. The sense of smell gives rise to the perception of odors, mediated by the olfactory nerve. The olfactory receptor cells are neurons present in the olfactory epithelium, a small patch of tissue in back of the nasal cavity. There are millions of olfactory receptor neurons that act as sensory signaling cells. Each neuron has cilia in direct contact with air. The olfactory nerve is considered the smell mediator. The axon connects the brain to the external air. Odorous molecules act as a chemical stimulus. Molecules bind to receptor proteins extended from cilia, initiating an electric signal. The primary sequences of thousands of olfactory receptors are known from the genomes of more than a dozen organisms. There are 7 helix transmembrane proteins, but there are no known structures of any ore. There is a highly conserved sequence in roughly 3/4 of all ores that is a tripodal metal ion binding site, and Susslich has proposed that the ores are in fact metalloproteins that serve as a Lewis acid site for binding of many odorant molecules. Crabtree, in 1978, had previously suggested that Cu is the most likely candidate for a metalloreceptor site in olfaction, for strong-smelling volatiles which are also good metal-coordinating ligands, such as thiols. Zhuang, Matsunami and Block, in 2012, confirmed the Crabtree-Susslich proposal for the specific case of a mouse ore, MOR 244-3, showing that copper is essential for detection of certain thiols and other sulfur-containing compounds. Thus, by using a chemical that binds to copper in the mouse nose, so that copper wasn't available to the receptors, the authors showed that the mice couldn't detect the thiols. However, these authors also found that MOR 244-3 lacks the specific metal ion binding site suggested by Susslich, instead showing a different motif in the EC2 domain. When the signal reaches a threshold, the neuron fires, sending a signal traveling along the axon to the olfactory bulb, part of the limbic system of the brain. Interpretation of the smell begins, relating the smell to past experiences and in relation to the substance emitted. The olfactory bulb acts as a relay station connecting the nose to the olfactory cortex in the brain. Olfactory information is further processed and projected through a pathway to the central nervous system, which controls emotions and behavior as well as basic thought processes. Odor sensation usually depends on the concentration available to the olfactory receptors. A single odorant stimulus type is typically recognized by multiple receptors, and different odorants are recognized by combinations of receptors. The patterns of neuron signals helping to identify the smell. The olfactory system does not interpret a single compound, but instead the whole odorous mix, not necessarily corresponding to concentration or intensity of any single constituent. The widest range of odors consists of organic compounds, although some simple compounds not containing carbon, such as hydrogen sulfide and ammonia, are also odorants. The perception of an odor effect is a two-step process. First, there is the physiological part, the detection of stimuli by receptors in the nose. The stimuli are processed by the region of the human brain which is responsible for olfaction. Because of this, an objective and analytical measure of odor is impossible. While odor feelings are very personal perceptions, individual reactions are related to gender, age, state of health, and personal history. Common odors that people are used to, such as their own body odor, are less noticeable to individuals than external or uncommon odors. This is due to habituation. After continuous odor exposure, the sense of smell fatigues quickly, but recovers rapidly after the stimulus is removed. Odors can change due to environmental conditions. For example, odors tend to be more distinguishable in cool, dry air. Habituation affects the ability to distinguish odors after continuous exposure. The sensitivity and ability to discriminate odors diminishes with exposure, and the brain tends to ignore continuous stimulus and focus on differences and changes in a particular sensation. When odorants are mixed, the conditioned odorant is blocked out because of habituation. This depends on the strength of the odorants in the mixture, which can change perception and processing of an odor. This process helps classify similar odors as well as adjust sensitivity to differences in complex stimuli. For most untrained people, the process of smelling gives little information concerning the specific ingredients of an odor. Their smell perception primarily offers information related to the emotional impact. Experienced people, however, such as flavorists and perfumers, can pick out individual chemicals in complex mixes through smell alone. Odor perception is a primal sense. The sense of smell enables pleasure, can subconsciously warn of danger, help locate mates, find food, or detect predators. Humans have a surprisingly good sense of smell correlated to an evolutionary decline in sense of smell. Humans' remarkable sense of smell is just as good as many animals, and can distinguish a diversity of odors approximately 10,000 scents. Bushdie et al. Reported, however, that humans can distinguish about 1 trillion odors. This is because of the retronasal route in humans to increase sensation. However, animals such as dogs show a greater sensitivity to odors than humans, especially in studies using short-chained compounds. Higher cognitive brain mechanisms and more olfactory brain regions enable humans to discriminate odors better than other mammals despite fewer olfactory receptor genes. Different categorizations of primary odors have been proposed, among others this, which relies on 7 primary odors: musky, perfumes, aftershave, putrid, rotten eggs, pungent, vinegar, camphoraceous, mothballs, ethereal, dry cleaning fluid, floral, roses, pepperminty, mint gum. Although recently progress has been made, the idea of primary perceptions is disputed, and more so, probably the concept of primary odors. Analysis: the ability to identify odors varies among people and decreases with age. Studies show there are sex differences in odor discrimination. Women usually outperform men. Pregnant women also have increased smell sensitivity, sometimes resulting in abnormal taste and smell perceptions, leading to food cravings or aversions. Deficits in smell also increase with age as well as the prevalence of taste problems. Chronic smell problems are reported in small numbers for those in their mid-20s, with numbers increasing steadily with overall sensitivity beginning to decline in the second decade of life, and then deteriorating appreciably as age increases to over 70 years of age. In Germany, the concentrations of odorants have since the 1870s been defined by olfactometry, which helps to analyze the human sense of smell using the following parameters: odor substance concentration, intensity of odor, and hedonis assessment. To establish the odor concentration, an olfactometer test is used, which employs a panel of human noses as sensors. In the olfactometry testing procedure, a diluted odorous mixture and an odor-free gas are presented separately from sniffing ports to a group of panelists, who are housed in an odor-neutral room. They are asked to compare the gases emitted from each sniffing port, after which the panelists are asked to report the presence of odor together with a confidence level such as guessing, inkling, or certainty of their assessment. The gas diluting ratio is then decreased by a factor of 2. The panelists are then asked to repeat their judgment. This continues for a number of dilution levels. The responses of the panelists over a range of dilution settings are used to calculate the concentration of the odor in terms of European odor units. The main panel calibration gas used is butan-1-ol, which at a certain diluting gives 1 OUA per cubic meter. General survey. The analytic methods could be subdivided into the physical, the gas chromatographical, and the chemosensory method. When measuring odor, there is a difference between emission and emission measurements. Emission measurement can be conducted by olfactometry using an olfactometer to dilute the odor sample. On the contrary, olfactometry is rarely used for emission measurement because of the low odor concentrations. The same measuring principles are used, but the judgment of the aerosol happens without diluting the samples.